A finger bone. That is what it took to discover an entirely unknown branch of the human family. Not a skull, not a tool, not a grave. A fragment of bone, no larger than a small stone, pulled from a cave in Siberia in 2008. And for 2 years, no one knew what they were holding. The Denisova Cave sits in the Altai Mountains of southern Siberia, a site that had already yielded Neanderthal remains when excavators reached a deeper sediment layer. From that layer came a juvenile finger bone, cataloged and sent to the Max Planck Institute for Evolutionary Anthropology in Leipzig. The researchers expected to confirm a Neanderthal or perhaps an early modern human. What the mitochondrial DNA returned in 2010 was neither. The sequence was distinct enough from both Neanderthals and modern humans to indicate a separate population, one that had not been named, had not been formally described, and for which almost no physical evidence existed. The cave gave them a name, Denisovans. To understand why that discovery unsettled the field, consider what the scientific consensus looked like before it. The dominant model held that archaic human diversity in Asia was relatively limited. Homo erectus had spread across the continent far earlier, Neanderthals had occupied Europe and Western Asia, and modern humans had migrated out of Africa and replaced both. The Altai region, in this picture, was peripheral, a crossroads, not a center. What the Denisovan DNA revealed was a population that had ranged across an enormous territory during the Middle to Late Pleistocene, from roughly 200,000 to approximately 32,000 years ago. That range was not established by finding skeletons across the continent. It was established primarily by looking at the genomes of living people. Roughly 5% of the DNA in Melanesians, Aboriginal Australians, and Filipino Negritos derives from Denisovan ancestry. In mainland Asians and Native Americans, that figure drops to around 0.2%. One study placed the highest Denisovan ancestry in Oceanians at approximately 2%, with most East Asian, South Asian, and Native American populations sharing similar smaller amounts of around 0.1%. The pattern implies that Denisovan populations were not confined to a single location. They were distributed across Asia, and modern humans interbred with them more than once, in more than one place. The physical record, by contrast, remained almost invisible. For years after the 2010 announcement, the confirmed Denisovan fossil inventory amounted to a finger bone, a few teeth, and a partial lower jaw found on the Tibetan Plateau at a site called Xiahe Cave. That mandible, known as the Xiahe Mandible, was identified not by DNA, the bone had not preserved it, but by ancient proteins extracted from its enamel. Protein analysis placed it within the Denisovan lineage. Its estimated age was at least 160,000 years, meaning Denisovans were living at high altitude, above 3,000 meters, long before modern humans arrived in the region. Then came the detail that changed the frame of the entire investigation. In June 2025, mitochondrial DNA and autosomal proteins extracted from the Harbin cranium, a large, massively built skull recovered in Manchuria and previously assigned to a proposed species called Homo longi, confirmed that it represented a Denisovan individual. The Harbin skull is not a fragment. It is one of the best preserved archaic human crania in the fossil record. Suddenly, researchers had a face. That face was low and long, with heavily developed brow ridges, wide eye sockets, and a large mouth. The Denisovans lacked a chin, as Neanderthals did. Their nose was larger than a modern human's. Their molars were larger than those of either Neanderthals or modern humans, more reminiscent of far earlier hominins. Yet their cranial capacity fell within the modern human and Neanderthal range, suggesting a brain of comparable size housed in a distinctly archaic skull. The same month, a separate study proposed that remains from 6 additional sites in China, including material associated with the roughly 1-million-year-old Yunxian man, may belong to the broader Homo longi lineage connected to the genetically confirmed Denisovans. Other researchers have placed specimens, including Penghu 1, a jaw fragment recovered from the seafloor between Taiwan and mainland China, within a proposed species called Homo joloensis, which some treat as encompassing Denisovans. These proposals remain contested. No formal species name for Denisovans has been officially established, and the question of whether they constitute a separate species or an archaic subspecies of Homo sapiens has not been resolved. The genetic picture is more complex still. About 17% of the Denisovan genome recovered from Denisova Cave appears to derive from admixture with the Altai Neanderthal population. Approximately 4% of that same genome comes from a lineage that diverged from the Denisovan, Neanderthal, and modern human lines more than 1 million years ago, a population consistent with Asian Homo erectus. And in 2018, a single individual from Denisova Cave was identified as a first-generation hybrid, a female with a Denisovan father and a Neanderthal mother, now referred to informally as Denny. The cave that gave Denisovans their name was, at various points, also a meeting place between at least two distinct archaic human lineages. Two broad explanations compete for how Denisovans could have left such a strong genetic signal across such a wide geographic range while leaving so little physical trace. The first explanation holds that the fossil record is incomplete by accident. Bone preservation requires specific conditions. Acidic soils, hot climates, and time itself destroy organic material. Much of the territory Denisovans may have occupied across Southeast Asia and the Pacific margins is geologically hostile to bone preservation. The Tibetan Plateau specimens survived in part because of altitude and cold. The Denisova Cave specimens survived because cave environments are unusual preservers. The absence of fossils across the rest of the proposed range may simply reflect what the ground keeps and what it destroys. The counter-evidence to comfortable acceptance of this explanation is the admixture data itself. The concentration of Denisovan ancestry in island Southeast Asia and Oceania, regions far from the Altai, implies sustained contact between Denisovan populations and the ancestors of modern Oceanians. For that contact to have produced the observed signal, Denisovans must have been present in numbers in or near the regions where those modern populations later developed. That is a large geographic claim supported by genetics and not yet confirmed by a single fossil from those areas. The second explanation, which several researchers have raised, is that what is currently grouped under the label Denisovan may not be a single homogeneous population at all. The genetic differences between the Altai specimens and the admixture signal in Oceanian genomes are substantial enough that some analyses suggest at least two distinct Denisovan populations, possibly more, separated by geography and time. If that interpretation is correct, the category itself may need to be divided. The physical remains so far recovered may represent only one branch of a more diverse radiation. What the Denisovan case exposes is a fundamental asymmetry at the center of paleoanthropology. For most of the 20th century, the field built its understanding of human prehistory almost entirely from bones and stone tools. Both are rare, fragmentary, and geographically uneven. Ancient DNA, developed as a practical tool only in the past 2 decades, can recover population history from material that leaves no visible trace in the ground. The Denisovans existed only in genetics for years before they existed in any recognizable physical form. That is not a solved problem, it is a changed method, with consequences still being measured. A related fossils question opened in September 2025 when a study proposed that material from sites across China, including Yunxian man, may connect to the Denisovan lineage. If those attributions are confirmed through protein or DNA analysis, the Denisovan story extends back close to 1 million years and encompasses remains that were already in museum collections, already debated, already assigned to other categories. The history of an entire archaic lineage may be sitting in existing collections, waiting to be reread with different tools. Return to the cave. Return to the finger bone, too small to tell at a glance what lineage it carried, extracted from a layer of sediment shared at different times by Neanderthals and by Denisovans, and by at least one individual who was both. The Harbin cranium now gives that lineage a face. The Xiahe Mandible places them at altitude 160,000 years ago. The genomes of living people in Oceania carry evidence of contact no fossil has yet been found to document. The precise question the evidence cannot yet answer is this: where and under what conditions did modern humans and a Denisovan population in or near island Southeast Asia meet often enough to leave a 5% genetic signal in the descendants of that encounter? And why has not a single confirming fossil been found anywhere along that road? The finger bone pointed outward across a continent. The signal is clear. The source is still missing. The unsolved vault. Every case leaves something behind.